हेलो हाँ अच्छा लिंक
आगे लो.
अच्छा अच्छा हाँ
Not this 4 only you have to go.
দেখছি ব্যাপার করতে পারি কিনা দেখছি
4 8th slide.
যা শুরু করব ঠিক আছে ও ওকে ফাইন
Ah. Yes.
वही तो लिंक भेजा था हमें इस पे भेजा है तो लिंक पे डाला है ना ईमेल में भेजा नहीं ना
Okay. Okay. So good afternoon, everyone. Welcome to this, and we will continue from where we left last time. So we are talking about protein structure determination using NMR. So the basic strategy has been explained, which is based on using TOSY and NOSY experiments, which are both proton-based experiments. And we say you get various kinds of distance constraints from the NOSY and coupling constant constraints from the COSY. The coupling constants will yield you torsion angle constraints. Put together, you define a potential function and try to minimize this potential function to get an acceptable structure. And after you got the structure, of course, you use in either of the two algorithms, distance geometry algorithm or the restrained molecular dynamics algorithm. Either way, you can calculate the structure. Once you calculate the structure, you have to validate it. You validate it by putting in all your torsion angles which you got in our structure, the 5-side torsion angles for all the individual amino acid residues on the Ramachandran map, and then check whether it is in an acceptable region or in a non-acceptable region. So here, this Ramachandran map is showing you a general statistics of a large number of structures put in on the Ramachandran map. And all these blue dots are the values for the individual amino acids in different proteins all put together to give validity to the Ramachandran map itself. So when you see that various different structures when you take and you put them onto the Ramachandran map, they all satisfy the constraints which are given by the Ramachandran map. So therefore, when you determine a new structure, you should check the correctness of that structure by putting in those 5-side values in the Ramachandran map. Okay, so we continue from there, and typically when you do the calculations using NMR, there are various kinds of NOEs. So NOEs, as I said, give distance constraints. There are NOE constraints can be classified in different types. There are intra-residual correlations. Then there are sequential correlations. Then there are medium range correlations. These are all listed here, and long range correlations. Long range NOEs, which are greater than 5 residues apart. Medium range is less than 5 residues apart. Sequential means near neighbor. So, and then you have the intra-residue within the same residue. And then, so therefore, you have, this is for a particular data, for a particular protein, you have a total of 1,665 constraints. So, and then you have 79 dihedral angle constraints, which are obtained from the coupling constants. This gives you the phi torsion angle. And then, of course, we have these other kinds of energies, which are bonds and angles and impropers and all of that. And finally, you end up with this kind of a statistics here, which is the most important thing. Here we see all the values and how many of these distance, I mean the 5-side angles, they are in the allowed regions in the Ramachandran map. How many are in additionally allowed, generously allowed, or how are disallowed regions? See, for example, in this structure, most favored region, 74.5% of the things are in the most favored regions. Then there are additionally allowed regions. Which are additionally allowed? These are all these yellow ones there. Okay, the real hot ones are these red and the brown kind of thing, but the additionally allowed are in the yellow region, or sometimes to a section in the brown region also. So that is additionally allowed. So you have, that is also allowed region, but of course it is slightly higher energy. The population will be somewhat smaller. So you have this 23% of those, and the generously allowed regions. This is the yellow, this is the brown, this is the yellow is the generously allowed. Disallowed region is very, very small. So you see only 0.5% are in the disallowed region. What that means is that this is a good structure, acceptable from the structural validity point of view. So this is the kind of statistics that generally presents when we determine structure, deposit your structure in the PDB or in the BMR, which are two databases where you deposit all your structures. This is the data one has to provide. Okay, so now we go into more complicated proteins, and then there are little bigger proteins, and the NOE spectra are also very complex. Now we see the 2D NOE spectrum, we compare this with the previous one which I showed, and here you have so many peaks, thousands of peaks, and they are in such close proximity, and there are many of them are overlapping also. So when they overlap here, then how do you actually quantify these peaks? Then how do you quantify the intensities of these peaks, and how do you estimate the distance constraints? And that becomes a limitation. So when you go to larger proteins, so this becomes a limitation that the two-dimensional NOE spectrum is not sufficiently well resolved. And then of course, this is again, this is at a 500 megahertz NMR spectrum. So what do we do to increase this resolution? You go to higher magnetic field. Maybe you can go to 800, 900, and today you have also 1.2 gigahertz. However, when you go to such large magnetic fields, you also have additional complications due to chemical shift anisotropy and line broadening because of that, and so on and so forth. So nevertheless, by and large, and they are also very expensive, not easily accessible, not easily available. So, but by and large, 700, 800 megahertz spectrometers are pretty much available in many places. So one can go to that. So you go to higher field magnetic field, then the dispersion will increase a little bit more. So you can go to somewhat higher molecular weights, and this is actually a lysozyme spectrum. This is lysozyme. This is 130 residues. This also is sufficiently complicated. So, but you will come across these situations when your aim is to study all proteins in your system. That 100,000 proteins are there, and they are all varying molecular sizes. So therefore, you will invariably come across such kind of situations. So what do we do there? Then of course, well, try and use other nuclei. So far, we are focusing only on the proton-proton COSY spectrum or proton-proton NOESY spectrum, but you can also use heteronuclei like carbon-13 or nitrogen-15. But then these are not naturally abundant. Carbon-13 and nitrogen-15 are not naturally abundant isotopes. So N15 is 0.37% in natural abundance. Okay, 15N is 0.37%, and carbon-13 is 1.1%. Natural abundance is so small. So therefore, what do we do? So then you cannot do it in natural abundance, these experiments. So then you will have to get uniformly labeled proteins, uniformly enriched by nitrogen-15 or C13. This requires molecular biological tools. You have to express the proteins inside cells, bacterial cells, which are fed by this C13 labeled glucose or N15 labeled ammonium chloride as the sole source of carbon and nitrogen. And when the cells take up these, and then they incorporate these isotopes inside these proteins, which they synthesize, then you isolate the protein from there and purify the proteins and so on. So that is the molecular biology step involved there. These are called as recombinant proteins. So recombinant proteins are, you have to introduce the gene corresponding to the particular protein of interest in a kind of a plasmid, and then the plasmid is introduced in a bacterial cell. Mostly you use E. coli cells there, and you grow the cells under this under the conditions of minimal media, which contains N15 labeled ammonium chloride or C13 labeled glucose. And then when the cells grow, you actually then destroy this, lyse the cells and extract the protein and purify. Having got that, having obtained that, now you can record heteronuclear experiments, heteronuclear two-dimensional experiments. So here it is the so-called HSQC. We have actually talked about the HSQC earlier in the previous set of lectures. We have seen this is heteronuclear correlation. Proton on this axis, amide proton on this axis, and here it is a nitrogen 15. Nitrogen 15 proton correlations. What does it tell you? That this is the proton N15, NH. This is the backbone, okay, HN. So proton and nitrogen 15 correlate. These are one bond correlations. So there will be as many NHs as there are amino acids. The protein will live in the side chains. So there's the backbone, there are as many NHs as there are amino acid residues. Therefore, and you will get here one peak per amino acid residue. So in this correlation map, this is basically a kind of a cross peak region in the homonuclear spectra. So this spectrum contains exclusively correlations only. Therefore, there is no such thing as a diagonal here. So you have only correlations, amide protons on this axis and nitrogen 15 on this axis. Okay, you can also have a C13 on that axis. So this is called a heteronuclear single quantum coherence spectroscopy, and you have one peak per residue in this spectrum. And the side chains also will appear if there are glutamines and asparagines. They will also appear. Those peaks will appear in pairs, as I discussed in the previous lectures. Therefore, this spectrum is known as the fingerprint of the protein. It represents the amino acid sequence. How many amino acids are there? Therefore, you have counted the number of peaks here. You must have as many peaks as there are in your amino acid residues in your protein, except, of course, proline. The proline does not have an NH along the backbone. Therefore, accepting proline, you count all the residues, you must have as many peaks as there are number of residues. Sometimes you may not see a peak for the N-terminal residues because the NH proton of that exchanges very rapidly with water and it may get broadened out and you will not see it. It will be under the water and you may not see it. But other than that, you will see most of the peaks. And even there, if you go to lower pH values, you might see all the peaks. So this is a very beautiful, well-resolved spectrum of a protein which is called as FKBP. You can count here all the residues are indicated here. Of course, this information has come after the assignment process. This is only I've taken to show you that okay, you will get one peak per residue. And here also there are distinct chemical shift regions. Here you see the top portion is more of glycines and threonines, and once you come here, you will get all mixtures of all of those things. You have the arginines, leucine. The distinct peaks are the glycines which appear on the top. The threonines also appear here in this area. Serines and threonines appear here. But other than that, of course, there is not so much of residue-wise chemical shift characteristics here. So all of them appear anywhere in this central region. But they will be well resolved. Okay, now here is an example of another protein which is HIV protease. It is a dimer of HIV protease with a linker in the middle which is GGSSG. The HIV protease 1, this is a 99 residues long. So and then you have, but there is a symmetry there because you have GGSSG link which is very flexible. So this HIV protease and this two domains are identical. So you will see only peaks from corresponding to the monomer. And of course you will see peaks of the GGSSG. And that is the linker. So you will see here those peaks are present here in this, all the linker peaks are present, all the glycines are present here, therefore the linker peaks are also present here. And this is the area which is quite crowded, and this is a situation where the protein is in a denatured state. Okay, HIV protease is denatured using guanidine hydrochloride, and then you are recording a spectrum in the denatured state. Even there, of course, you can see all the peaks pretty well resolved in this case. So the N15 actually is a very useful nucleus because it has a very good dispersion. And this area is blown up here in this region, and you can see all the individual amino acid residues have been identified. So you can do the heteronuclear experiments which provide you a better tool to go to larger systems and extract additional information than what the proton alone can give you. Okay. Now there are also other things which you can do. That is the proton N15 HSQC was one thing. But you can also have proton carbonyl correlations. Okay. So this is the aromatic ones with the 2D HMCO, and you can also have that sort of a thing. Then here you have correlation from, this is the HSQC. This is the typical N15 HSQC. This is These chemical shifts are of the nitrogen 15. These are chemical shifts of the carbonyl. Okay, these are chemical shifts of nitrogen 15, and there it is amide proton. Okay, the experimental sequence may have a particular kind of a sequence, but you can extract whatever information you want to, whichever projections you take and obtain whatever information you want to get. In this experiment, this was labeled as 2D HMCO, but you actually get two kinds of data sets. So one is you have the amide proton to the nitrogen 15 correlation. You see the chemical shifts here, 104 to 120, and these are 15 chemical shifts. On this side, you see what chemical shifts, carbonyl chemical shifts. These are 160 to 184. Typically, the carbonyls are concentrated in this area around 168 to 180. So you will see these carbonyl chemical shifts are also pretty well dispersed, and the lines are also quite sharp. Carbonyl lines are also pretty sharp, and therefore you can also use this sort of a correlation. And you make use of this dispersion of the spectra on the basis of heteronucleus, and then you can use this to go to larger and larger molecular systems. Okay, so now what you do is you go one step further. So the first step was to go to higher magnetic fields to improve the resolution. The second step was to use heteronuclei to improve the dispersion, to get additional nuclei, additional information. Now what you do is you combine the proton-proton with the proton X nucleus heteronuclear correlations. So this bottom one is the proton-proton correlation here. The top here is the proton nitrogen 15 correlation. Now what you do is for the same system. Okay, this is the schematic to indicate how this experiment, you extend the dimension of the experiment to go from 2 dimension to 3 dimension, then how it will be helpful. Okay, I will explain this first, what is the purpose, and then of course we'll go to the multidimensional sequence which we have investigated earlier as well. Now here is a particular situation you consider. At one particular amide proton chemical shift, and on this side you have the full aliphatic chemical shift. Here is only the HN, the amide proton. And on this side you have the full proton, all the entire proton region. You are seeing so many peaks there. You can see actually 10 peaks there. 3 for 4 plus 3, 10 peaks are there. So any amide proton does not produce 10 peaks. In any amino acid residue, you don't have such kind of a correlation. So many proton correlations are not there in the 2D HSQC or in the 2D NOSI or 2D COSI, whichever ones. These are too many. What does that mean? This implies that at that particular amide proton chemical shift, there are more than one amino acid residue. There is a degeneracy of the amide proton chemical shifts. And because of the degeneracy, peaks corresponding to the different residues are appearing differently, and this degeneracy is not showing up in the other aliphatic protons. And they are separated, but the amide proton is the same for all of those. Maybe there are 3, 4, we don't know how many are there, but they are all showing this. Now what you do is you do amide proton N15 HSQC spectrum, and now you see that on the same system you see that there are 3 peaks there. What does it mean? At that particular amide chemical shift, there are 3 nitrogen 15s, which means there are 3 amino acid residues whose amide protons are overlapping. So at least 3 are there. If there are further overlaps there, we don't know. That is, there are at least 3 amino acid residues. Now what you want to do is to improve the resolution in your spectrum. You pull this 2-dimensional spectrum in a 3rd dimension using the N15 chemical shift. N15 chemical shift, how many are there? There are 3, 1, 2, 3. So when you put this on the 3rd axis, nitrogen 15 axis, these 10 peaks will get distributed on 3 planes in the 3-dimensional spectrum. So you get a cross peak here. There is one plane which is 3 peaks. Another plane, there are 4 peaks. Another plane, there are 3 peaks. So total 10 peaks are now stretched out into the box in the 3-dimensional sense, and all these peaks are now separated out. Now on the basis of the individual chemical shifts, you can identify what that residue could be. You have seen what sort of expectations will be there for different amino acid residues along the spin systems. Now, to get further resolution on these ones, you can also combine that with carbon 13 to disperse this further on the basis of carbon 13 chemical shift. You can make use of carbon 13 chemical shift as well. When you do that, these 4 peaks, for example, will get distributed in 4 different planes in a 4-dimensional spectrum. Because what you do here is you take the particular N15 chemical shift and generate a 3-dimensional spectrum where you have amide 1 here, and another all aliphatic protons here, and on this axis you have the carbon 13. So all of these four peaks are separated on the basis of the carbon 13 chemical shift. Okay, so therefore this allows you to improve the resolution in your spectrum, dispersion in the spectrum. This is done by what is called as multidimensional NMR techniques. Multidimensional NMR is schematically shown here. So you have this is the time axis. On this axis, this is the time here. So you have the preparation period, evolution time T1, and you have the mixing time 1, M1, the evolution time T2, mixing time 2, and then you continue like that. If you have n times here, then you generate a n plus 1 dimensional spectrum, because there's a Tn plus 1. But if n is equal to 2, then n is 3, therefore you will have a 3-dimensional spectrum. If n is equal to 3, then you'll have a 4-dimensional spectrum. So you can actually design these various kinds of mixing sequences here. One mixing sequence, the second mixing sequence, then you have the evolution periods T1 and T2, and you detect finally the T1 at the end, this detection period. So these T1 and T2 are systematically incremented, like in the 2D experiments, independently incremented. So you generate 3 time variables, T1, T2, and T3. Then you will have to do a 3-dimensional Fourier transformation to get a 3-dimensional spectrum. Okay. So now here is an example of another kind of an example, which is a 2-dimensional spectrum all right here, but we are using 2 types of mixings. So we start transfer information. This is called as HSQC nosy. What you do is you make use of both these combinations. In a particular 2D experiment, you perform an HSQC until here you have the HSQC spectrum. Okay, so until here you have the HSQC spectrum until this period. Transfer here, then transfer there, and refocus here. And after that, there's a magnetization. You put it along the z-axis. You mix it with the nosy mixing part. So you have the here it is the HSQC part, and then you have this nosy mixing. Therefore, you get a HSQC nosy spectrum. Here there is only 2 independent variables, T1 and T2, and therefore you get a 2-dimensional spectrum. But you have the peaks separated out on the basis of the N15 chemical shifts as well. So what is present here? This will be the N15 chemical shift or the C13 chemical shift, whatever you might have. And this will be the proton chemical shift. This is the amide proton chemical. This is the proton chemical shift. So you will have a 2-dimensional spectrum, but it is having both of the combined. That means the nosy spectrum which is there is now spread out, or the HSQC spectrum is pulled apart on the basis of the nosy interactions. And typically one uses tau is equal to 1 by 4j. This is the typical HSQC scheme what you have studied it earlier. Okay, now this is the 3-dimensional experiment of the same type. 3-dimensional nosy HSQC. So what you do is first you have the nosy part, the T1 mixing time. So until here it is the nosy. And then you do an HSQC. Okay. Then you have the HSQC experiment and detect the signal here as T3. So you have 3 time variables, T1, T2, and T3. T3 is proton and T2 is nitrogen 15 or C13, that's here. And the T1 is proton. So what you do is you generate the nosy mixing here, that you excite the protons, amide protons, and then you relay the information to other protons. Okay, and then you can transfer that information to the heteronucleus. Here, you can transfer it to the heteronucleus and label the heteronucleus here and transfer it back to the proton and refocus here, and you can collect the data here. So therefore, this is systematically incremented, this is systematically incremented, and this is the detection period. So you get a 3-dimensional experiment where you have made use of the nosy mixing as well as the HSQC mixing. And of course, when you are doing here, you can decouple carbon 13 and 15. So here is an illustration of that one. So you have on this axis, this is a typical 2-dimensional spectrum. You have proton N15 correlation spectrum, proton on this axis and N15 on this axis. And then you have the nosy. So each of these peak here, if you take these various sections you generate proton NOEs along this axis. See, every peak here generates the protons along this axis. And these are the NOEs. So from each of these amide proton, you are generating the NOEs to the various aliphatic protons. And these residues are separated along this axis using the N15. What happened?
One he was big.
And so each of these peak is now representing one amino acid residue. Okay, this is the HSQC spectrum, remember. This is the N15 proton HSQC spectrum. Therefore, each peak is representing one amino acid residue. And that is the same thing here. The projection down here, this is the same thing there. And so from each of these peaks, you go on the vertical axis, you generate the entire NOEs. So you have, now the NOEs are now spread out in a box. And therefore you will have a very high resolution in your spectrum. Okay, so this is a typical, you can see this is the proton axis on this axis, nitrogen 15 here, and proton axis on this axis also. This is quite clear. So in the cuboid, you have all the peaks distributed on at different places. Okay, now this is the same experiment, and it is now one particular plane of this cross section. You see so many things are there, but that means at that particular N15 there are many residues. There are N15 there are many residues, and all of these have different NH proton chemical shifts, and they are showing individual NOEs to the different aliphatic protons. So these may be nearest neighbor or the long range or the short range or whatever. So NOEs to the aliphatic hydrogens, and these ones are NOEs to the individual amide protons there. So these are structural information. If you recall back what we said, so these ones are having structural information with regard to the helices, and here you will have the information with respect to the beta sheets and so on, so forth. So all of these will be from. Okay, so therefore you see each strip contains NOEs from one NH group to all other hydrogen atoms close by. And each plane represents one N15 chemical shift. That means that N15 chemical shift, there are so many different NH protons. There is a degeneracy of the NH protons at that particular N15 chemical shift. Okay, so now you go, yes. So now we go over to another set of experiments where we make use of both nitrogen 15 and C13. And these are called as triple resonance NMR experiments. This requires both carbon 13 and nitrogen 15 labeling. There are a large number of experiments. I have listed only a few of them which are most commonly used. And we will discuss a little bit of these ones to see what is the information content in each of these spectra. Schematically, if you see this, all these was showing the schematically the kind of information that is. This can make video. And these are showing the schematically the transfer of magnetization, transfer of information, or the labeling pathway, how the magnetization is transferred in the pulse sequence. Okay, so let us take for example the HMCA. So this is a three-dimensional triple resonance experiment. Why you say triple resonance? Because you have the amide proton channel, the nitrogen 15 channel, and the carbon channel. All the three. All the red ones are the ones which indicate the nuclei which are observed in the experiment, either in any one of the directions, any one of the dimensions. If it may be F1, F2, F3. F3 is by and large only the amide protons. F1 and F2 can be different ones. And in this situation, the amide magnetization starts from the amide proton. That means you excite the amide protons initially, and then it is transferred by the inept sequence to the nitrogen 15. And from the nitrogen 15, nitrogen 15 is labeled there, frequency label that constitutes the T1 variable. And then from the nitrogen 15, it transferred to the C alpha of two residues. It's same residue and also the previous residue. So if this residue is I, this is I minus 1. So you transfer to the previous residue, and then you label the C alpha. So that becomes the frequency labeling of this carbon. And from that one, you come back to the nitrogen 15. And now you don't have to label that, you already labeled it. And from nitrogen 15, you come back to the amide proton and detect the amide proton. Therefore, this is a three-dimensional spectrum wherein you will have amide proton on the F3 axis, nitrogen 15 on F1 axis, and C alpha on the F2 axis. You can also turn it around and say, okay, I want to label nitrogen 15 when I come back, not while going. That means in which case you label the nitrogen 15 after you come back from the C alpha. So this way you can either have F2 as nitrogen 15 or F2 as carbon 13. Either way one can do it. Now that is a extra. Now in this particular case, therefore what we will see here, you will see correlations from the amide proton to the nitrogen 15 of the same residue and the C alpha of the same residue and the previous residue. So you will see those three correlations. Now if I look at the HMCO-CA, this is an experiment, this is the transfer process is different. And this will reflect in the pulse sequence. How you design your pulse sequence, it will show up in that one. So you start from the amide protons, excite the amide protons, transfer the magnetization to the nitrogen 15, to its own nitrogen 15, amide. Now from the nitrogen 15, you transfer to the carbonyl. You don't transfer to the C alpha straight away as we did it here. Here you can. How we could do that here? Because there is a coupling constant. All these are based on the coupling constants, right? So there is a 2-bond coupling and a 1-bond coupling. So in this case, we used both of the 2-bond couplings and the 1-bond coupling. And in this case, you are using nitrogen 15 to carbonyl. This is the 1-bond coupling. And from carbonyl, you don't label that there. From carbonyl, you go to the C alpha. That means it will go in one direction only. It goes to the C alpha of the previous residue. And you can label there. Then you come back to the carbonyl, come back to the nitrogen 15, then you come back to the proton for detection. So this is how the magnetization transfer happens in the individual experimental sequences. Of course, we will go through, show a little bit more in those details, but this is the schematic of the magnetization transfer scheme in the different three-dimensional triple resonance experiments. So we will be going back and forth on this, but this is a schematic of the coupling constants which are used in all of these experiments. The various coupling constants which are used in this are listed here. Notice the 1-bond couplings. The 1-bond couplings are the NH. This is the 90 to 95 Hertz. So that was actually you had to tune your delays according to the coupling constant. And it doesn't vary too much. 90 to 95 doesn't vary too much because these are all 1-bond couplings. And nitrogen 15 to carbonyl is 15 Hertz. And carbonyl to C alpha, this is 55 Hertz. And C alpha to its own proton is 140 Hertz. C alpha to C beta is 35 Hertz. And then this nitrogen to this C alpha, and that is 7 to 11 Hertz. Then this nitrogen, the 2-bond coupling to this C alpha is 4 to 9 Hertz. This shows a larger variation. And this, because the numbers themselves are very small, so this appears as a little bit larger variation. Okay, so this will require a little bit of the tuning there to be able to get a transfer of information there. 1-bond coupling, the variation is very, very little. In the 2-bond couplings, the variation is little larger, but that requires certain amount of tuning. So this is residue I and residue I minus 1. You have these coupling constants which can be used for optimizing your pulse sequences. Okay, so let's say for example one of these experimental sequence which I showed you, this is called as the constant time HMCO. We have discussed this earlier, but I will briefly run through it once more. So you start from, I explained in the schematic way how experiment is done. So here it is the proton axis. The experiment has obviously these channels there. This is the proton channel, the nitrogen channel, and both these are carbon channels. What does that mean? Are there two different channels? No. This is the same carbon channel, but you have written these two lines separately because of C alpha and CO, because you can apply selective pulses to these carbon C alphas and the COs without any problem. Because the C alpha chemical shifts are in the 50 to 60 Hertz or 45 to 55 Hertz range, chemical ppm. Okay, C alpha and the carbon side is about 45 to 55 ppm. Okay, and where are the carbonyls? Carbonyls we have seen 160 to 180. These are very far away. Therefore, it is no problem exciting either this region or this region. Therefore, even if the channel is the same, you can apply selectively pulses only to the C alphas or the COs, and that's why these are put on separate lines there. So the experiment goes in this manner. The transfer process is indicated here. So you start from the amide proton that is here. You transfer to the nitrogen that comes here, nitrogen to the C alpha. Then you label this nitrogen during this period. This is the constant time period. And during this nitrogen, nitrogen is labeled. And then you transfer to the C alpha that comes here. At this point, there is a C alpha. And it transfer happens to the C alpha of residue I and I minus 1, as I showed you earlier in the HMCO sequence, and you label the C alphas here. Therefore, the T2 period, the C alphas are labeled, and then from here you transfer it back to the nitrogen 15, and then this constant time period is, and now you are not doing any more labeling here. You already labeled the nitrogen 15 here, so you don't need to label that here again. So from the nitrogen 15, allow it to evolve and refocus some of the coupling constants and so on and so forth, and then you transfer it back to the amide proton at this point. Amide proton, this is anti-phase magnetization here, and I will not do the technical language just in this set of lectures, because we already did that in the previous case. So you have this from here, the anti-phase magnetization is refocused to in-phase magnetization of the amide proton, so that you can decouple then amide protons from the nitrogen 15 here. This is the decoupling. You observe the amide protons without any coupling to the nitrogen 15. Now, so the density operator is given here at this point. At this point, what is detected? What is the beginning of the detection period? The density operator is given here. So this is the amide proton chemical shifts. Okay, this is amide HYNX. This is of the residue I, and it has these various coefficients there. And these coefficients are responsible for the cross peaks in your spectrum. What do they depend upon? See, they depend upon, these determine the intensities of the various peaks. Okay, first of all, they define the chemical shifts. Chemical shifts, which chemical shifts? Okay, now you see here T2. Where is T2? T2 means this is the C alpha chemical shift of residue I. And here again T2, C alpha chemical shift of residue I minus 1, because I told you are going to label C alpha in this T2 period. Therefore, C alpha I minus 1 and C alpha I. These chemical shifts are there. Now, what is this here? T1. T1 indicates the labeling of the nitrogen. That is happening here. So T1, in the T1 period, you have the labeling of the nitrogen. Therefore, and of course, this is this is by coupling constant dependent T2, which means it leads to a splitting of this C alpha C beta due to the coupling of C alpha C beta. And these coefficients, and these are constant time dependent coefficients there. Okay, gamma 1 and gamma 2, these are proportional to the C alpha coupling constant, C alpha nitrogen coupling constant, and the time T, the constant time period T. So, and one bond, this is the one bond coupling constant, this is the two bond coupling constant. The gamma 1 has this combination there, gamma 2 has this combination there. So this determines the intensities of the individual peaks. So starting from a particular nitrogen I generate a correlation to the C alpha of I and C alpha of I minus 1. And this will be at and the T3 is what? T3 will be the amide proton of I, because I am coming back here to I. Therefore, T3 will be omega HNI. So this is the amide proton chemical shift. So therefore, all these three will appear along the three different axes in your three-dimensional experiment. What does it appear? On the F3 axis, if I were to draw this plane here, I probably also have a schematic over there. This is F3, this is F1, this is F2. Here I have the C alpha, here I have the N15, and here I have the HN. Okay, so this is the way the three-dimensional spectrum will look like. And all of these coefficients determine the intensities of the peaks. Now this is the schematic shown here once again. I've shown you that one. They explained that, but here the appearance of the spectrum is explicitly demonstrated there. You consider here 4 amino acid residues. You consider 4 different ones. So you have here the green, brown, cyan, and red, and an individual amino acid chemical shift, individual amide proton, for example, here you see two peaks along the C alpha. Therefore, C alpha of I and C alpha of I minus 1. That is indicated that I shown a strip. You take a strip here that is shown on this one. You will have at the N15 of I, you have F and the HN of I, you have the C alphas, one to its own and to the sequential I minus 1. Similarly, you have for the other ones also. And normally you will find that the self peak is a little bit stronger than the sequential peak. So therefore this is a sequential peak and this is a self peak. Self peak is slightly stronger than the sequential peak, and that's why we have shown it as a bigger circle. And this will be useful for obtaining sequential walkthrough because you established a correlation, you have established a sequential correlation. So you can go from one plane to another plane and find out your peaks, and then you can do a sequential walk along the polypeptide chain. So this is indicated here. The same four peaks which are there. So if I were to look at the green one, I have the self and the sequential. Now what I do is I search through the N15 planes, scan through the N15 planes and find out if I have a C alpha at this particular chemical shift. Which chemical shift? C alpha chemical shift. And I find that, okay, yes, at a particular plane we have this, and I have the self peaks here at the same chemical shift. And then of course at that point NH, I will also have the sequential peak to the next residue, I minus 2. And then again I search through the N15 chemical shift and find out if I have another C alpha at this point, this chemical shift. Then I see that I find here. So therefore this will be a self peak, and again a sequential peak, a self peak, and sequential peak. So therefore you see you can walk through the polypeptide chain, but you have to scan through the nitrogen 15 planes to find out exactly which nitrogen 15 plane has which C alpha. So therefore you will have to scan through it every now and then, find out where the peaks are, and you can walk through the polypeptide chain. And here there will be no ambiguity, because no question of peak not appearing, because these are all based on fixed coupling constants which do not vary. They are not dependent on the structure of the molecule. They are only dependent on the sequence. Therefore, the sequence variation is not very large. I mean, the coupling on variation is not too large in one bond and two bond couplings, and therefore you will see all of these peaks all the time. And you can go to larger molecular weights. And this is an experimental situation here. So you start from here, for example, you have the sequential correlation. This is the sequential peaks, the self peak and the sequential peak, and you go horizontally and you keep walking like this as I showed you there. You see, you can go identify various nitrogen 15 planes. See, you started, let's see, you start from the A19. You can either go from this way or you go from the top. The 90, A99, alanine 99, you have a self peak here, sequential peak there. Therefore, you search to the plane, you come to the self peak here. This is a residue 98 self, sequential peaks. Then when you go horizontally, you come to the self peak of 97, the sequential peak of 96, go here, the self peak of 96, then you have the sequential peak here, so you can walk along. So in this way, you can walk through the polypeptide chain without much problems. Okay, the backbone therefore gets assigned in this manner. So this is the schematic here, and this is the expansion of that. Okay, now HMCO is another experiment which is whose magnetization transfer pathway is indicated in this manner. This also I schematically explained to you earlier, HNCOCA, and here you start from the amide proton, go to the nitrogen 15, that is here, and from the nitrogen, this is the T1 period here. During this period of the T1, go to the carbonyl, from here you go to the carbonyl. Now you apply a pulse to the carbonyls, nitrogen 15 and the carbonyl, and so therefore the magnetization comes here in the carbonyl, and this is the residue I minus 1, carbonyl of residue I minus 1. And then during this period, you transfer from the carbonyl to, from here to here, you transfer to the C alpha. Transfer the C alpha residue I minus 1 to C alpha of I minus 1. You come here, C alpha of I minus 1. And you label the C alpha here at this point. So this is T2. Therefore, this is the independent time variable. Therefore, frequency labeling will happen for the C alpha. And then from the C alpha, you go back here to the carbonyl. As you came here, just as you came here, you go back here, carbonyl, and then you evolve further and then you transfer back to the nitrogen. From C alpha COI -1, you transfer back to the nitrogen here, and you transfer back to the nitrogen here, and then from nitrogen you go back to the proton here at this point and refocus and collect the amide proton chemical shift, decoupling the nitrogen. Therefore, you see, you have established a kind of a directionality in your sequential work. In the earlier case, you had the C alpha I and I -1. It was going on both directions. Now you are going to only one direction, C alpha of I -1 only. You are not going to the C alpha of I. Therefore, you are able to distinguish if there is any ambiguity in the previous situation as to which is the self peak and which is the sequential peak. This experiment will resolve that. You will identify exclusively the one which is sequential. Okay, so this is the same schematic here. You are only seeing the sequential peak for each NH proton at a particular Ni chemical shift. Okay, now this is another experiment which is called HMCO, and without having to go through the full details of this experiment, so this labels nitrogen amide proton on one axis, nitrogen 15 on another axis, and the carbonyl on the third axis, and therefore this is called as HMCO GP 3D experiment. So we need not go into the details except to mention that, okay, here you have the initial transfer to the nitrogen, and then transfer to the nitrogen, you have the carbonyl labeling here, the nitrogen labeling here, and okay, in this case the nitrogen labeling is happening at this point, not here. Nitrogen labeling is here happening at this point, the carbonyl labeling is at this point, and finally you are detecting the amide proton. And anybody who wants to work out the mathematics of this, then of course can work it out. Details, we need not go into those details, that will take quite a lot of time. But these all sequences are already well described in the NMR spectrometers. Libraries are there for all this, but you just click a button, okay, choose this pulse sequence, you have the pulse sequence. But it is important to know how these experiments work, what is the thing to be optimized, which pulse has to be optimized, what is the shaped pulse here. You see, these are all shaped pulses. All these kind of shaped things, not rectangular ones, they are shaped pulses. They are applied to the C alpha or to the CO, and those ones are indicated by the shaped pulses. Hard pulses are the black squares, those are hard pulses. And here you have the gradients. We have discussed the gradients also earlier, the Crusher gradients, various gradients are used. Okay. And that is the same experiment which is HMCO experiment. You can take projections on the different planes, on the different axes, you get different kinds of information, two-dimensional information. So if you have the proton, here it is a proton C13 chemical shift correlation, because you have taken a projection on the nitrogen axis. And here you have the proton C13 nitrogen correlation, C13 nitrogen 15 correlations, because you take a projection on the proton axis, projection down the proton axis. And here you have this is a typical three-dimensional spectrum. Proton on one side, proton here, and nitrogen 15 here, and carbonyl here. Okay, so this is the three-dimensional cube, and you take projections of this either down or like that or like that. Depending upon which projection you take, you get different kinds of two-dimensional spectra. See, that is extremely useful information what you have there. And this is a particular kind of projection here. You have in each NH strip there is one CO visible which belongs to the preceding residue. Okay, this is how you see. You take from the particular NH to the CO. That is what is seen in this particular plane at a particular N15. And particularly N15, there are so many residues, and for each NH, you are seeing a one CO peak. Okay, so therefore, clearly the degeneracy of these various things is lifted by making use of the different chemical shifts. Okay, now the HMCO-CB is another set of experiments which extends these things to further to C beta protons. Okay, C beta protons and C beta carbons. It goes to both. There are two versions of this experiment. One is called HMCO-CB, other one is called CBCA-NH. Okay, and what do they differ? So let's look at this here. If I look here, this is HNCA CV. It starts from the amide proton and goes to the nitrogen 15. And then from this nitrogen, it goes to the C alpha, as in the case of HNCA. Okay. And then from the HNC and from the C alpha, it goes to the C beta, same here and here. Okay. You label these also. You label both C alphas and C betas. And from C beta, you come back to C alpha, here and here. From C alpha, you come back to nitrogen, go here, and then come back to the amide proton for detection. So you have C alpha, C beta both present on one carbon axis. Nitrogen is present along the nitrogen axis, and the amide protons are present along the proton axis. In this experiment, the magnetization transfer pathway is slightly different. You are not starting from the amide proton. You are actually starting from the aliphatic protons. You start from the H alpha and the H beta. From the H alpha, H beta, you go to the C beta. From C beta, you go to the C alpha. Of course, there will also be transfer from the H alpha to the C alpha. Then from C alpha, you go to the nitrogen here. Same here, go to the nitrogen. And then you go to the amide proton for detection. So there is one way here. There is no going back and forth. In this case, you go one way. So what is the kind of information you get in this kind of experiment? See, this transfer process is indicated here. Okay. So what sort of peaks you will get? You will get amide proton on one axis. This will be the F3 axis. 915 on one other axis. And you get both C beta and the C alpha of residue I, and C beta and C alpha of residue I minus 1. See, you notice here N15 chemical shift is the same. Amide proton chemical shift is the same. But what you get? C alpha and C beta of residue I, C alpha and C beta of residue I minus 1. Therefore, in each strip, how many peaks we will get? We will get 4 peaks. So if I take the strip here, this is the HN of residue I. This is the particular N15 of residue I, and here I have the C carbon chemical shift. So I have here on the top C beta here, and bottom I have the C alpha. I will have 2 peaks here and 2 peaks there. So for each amide proton, I will have 4 peaks there in this. In the HNCA, I had only these 2, C alpha, self and the sequential. Here we are extending it further to the C beta. So you get the C beta chemical shifts also. That helps in identifying the individual amino acid, because of the chemical shift dispersion is there, making use of additional chemical shift dispersion. Therefore, identification of the spin system also becomes possible in this situation. So we need not go through the pulse sequence. I think you can skip this part. Those of you, but the slides are there. Anybody who wants to search, go through this, you can read this. But I will show you the experiment. These are the strips from the 3D experiment, various strips at different levels. So this is the analysis which is indicated there. Okay. Let's start from, see, the sequence is going from 915 to G29. So there is G29 alpha is here, and you are seeing the sequential to 28 alpha, because glycine doesn't have a beta, therefore it doesn't have anything on the top there. Okay, so you have here the beta alpha peak here, and you go to the C alpha of residue 28. And then you see 4 peaks, 2, 3, and 4. Okay, so you are seeing a peak from the S28 nitrogen to the C beta, to the carbon, to the beta of the S28 itself. This is alpha, this is the self peak. And what is this one here? This is the sequential peak. Okay, that is the sequential peak corresponding to this, from the G29 to G beta sequential. Self beta is not there. This is the sequential beta. So you have this one there. Okay, and then there is another weak one there. See, carbon is only one, right? So therefore you have the C beta of S28. Now from this one, From this 28, you have the sequential peak to 27 alpha. So 27 alpha goes you to the self of 27 and the sequential of 27. This is the 26. Sequential means 26, but this is the alpha, and you also have that in the beta there. So you see 27 alpha to this 27 beta. That is the self beta. The sequential 26 beta is not visible there. Okay, sometimes the beta sequential beta may not be visible. That can happen. Sometimes there is there, sometimes it is not there. So from this sequential alpha, you go to the 26 alpha self, and the sequential 25 alpha is here. Now from this 26 alpha, you see the 26 beta self and the 25 beta, which is a sequential. So you see, you can collect these in both the C alpha planes and also the C beta planes. So you come, if you look at the C alpha connections, go here, here, here. That's why they are given in different colors. Okay, 29 to 28, 28 to 27, 27 to 26, 26 to 25, 25 to 24, 24 to 23, 23 to 22, 22 to 21, 21 to self. And then of course you can go further. Same happens in the case of the betas. So you see here is self and beta, these are very close by there. See these two lines, the two sequential and the self are overlapping. So the 27 to 26 sequential peak is present here. Is that okay? Explicitly not. It is very close, it is overlapping there. So 27 to 26, 26 to 25, 25 to 24, 24 to 23, 23 to 22, 22 to 21, 21 to 20. So this is the way the sequential walk can happen. It can happen in both the C alpha and the C beta regions. Okay, now we extend this further to make it more directional. Okay, the transfer process, the CBCA-CONH experiment, this happens once again, it starts from here, the H betas, starts with the C betas, from C beta you go to the C alpha you go to the CO. What did you do earlier? You went from the C alpha directly to the nitrogen. Direct in the CBCA-NH, you went directly to the nitrogen, but we are not doing that now. So therefore, it now becomes a sequence, only one residue. It is not going to the two residues at the same time. So from the C alpha, you go to the carbonyl to nitrogen, nitrogen to this. Therefore, you will see only this correlation. Amide proton to the nitrogen 15 and to the C beta of I-1 and C alpha of I-1. You will see to C beta of I-1 and C alpha of I-1. So therefore, there is only one stretch of one set of peaks for each of the sequential peaks. And this is shown here. This is HMCA-CB, which I already explained to you in one case. Now here it is the CBCA-CONH. You see two peaks only in this line because we have C alpha or C beta, I-1 only, C alpha I-1 and C beta I-1 for each case. So you will have two peaks there, two peaks there, two peaks there. This is come from something else, and there are there's overlap here in this, two peaks there and two peaks there. So all the self peaks are sequential peaks to the sequentially connected I-1 residue are there. Okay, so this provides some directionality in the carbon walk along the polypeptide chain. Okay, so once you have this amide proton chemical shifts, C alpha chemical shifts, the C beta chemical shifts, and the NH proton chemical. So these are all the backbone chemical shifts. So you have, and then you can feed in this. The chemical shifts also have the structural information. Protein structural information is there on the chemical shifts also. And therefore, why not use the chemical shifts alone to calculate the structure of the protein? Do we need to really depend upon the NOEs? The NOEs are so complicated to analyze. They are so overlapping, and therefore it becomes a very difficult thing to do. But if we can do on the basis of chemical shifts, which also have the structural information, and that indeed was developed in two groups. They developed an algorithm. One is called CS23D, other one is called CS Rosetta. And these ones were published in the same year, 2008. And now you see, you put in this chemical shift information to calculate the structure of the molecule. And you see here, this is an example here. So this is the structure of the molecule, average structure. This is what you get on the basis of the structural data what you have got there. This is an average of all of those. Okay, so from the minimal data set, you can calculate the two other proteins. This is human ubiquitin there. This is chicken SH3 domain, and all the chemical shifts are introduced. From the chemical shift introduced, to calculate the structure using the Rosetta. So then you get a structure which is like this. Where the chemical shift information is not sufficient, this you had also in the case of noses. If the nosy input is not sufficient, then you will not get a proper defined structure. Where it is sufficient, you have a well-defined structure, and there are small variations in the individual structures because of the accuracies of the chemical shifts and the modeling procedures there. Therefore, you get an ensemble or a group of structures which are close, and then you can calculate an RMSD from those. And what is shown here is the structure calculated for these two proteins on the top, and what is plotted here is the RMSD with respect to the crystal structure. For these, the crystal structures are already known, they are known. So you calculate the RMSD, how much is the deviation, how much is the agreement between this calculated structure and the crystal structure. This because this establishes the reliability of the structure calculation. And you can see here this is pretty good. So is less than 1. This is the average value. This average value is large because of these large deviations at this point there. And that happens in the loop areas. By and large, in the loop areas, the structures are not as well defined. So if you consider the other ones there, you see this is so beautifully defined in this area. Most of the residues, the RMSD is less than 1, about around 1, which is not a bad thing at all. And in this case, it is even better there here. So in this structure, because the protein is well defined, protein is more stable, so you get a better structure calculation here from the chemical shift data alone. See, now the chemical shift data can be obtained relatively easily from this combination of different three-dimensional triple resonance experiments. You really don't have to go to the NOEs. NOEs are much more complicated to analyze because there are so many in number and some uncertainties in the intensity patterns. Therefore, these chemical shift-based calculations are pretty very useful, but this is giving you the backbone information. We are not yet gone into the side chains. The side chains will, of course, add to the complications. Now, you have to also obtain side chain assignments. There are separate experiments designed for the side chains. Now, these are based on carbon-carbon toxi. So, you see, this is called as HCCS toxi. The most common one is the HCCS toxi. So what you do here is you start from any particular aliphatic proton, you transfer the magnetization to the carbons, and then you do a toxi on the carbons. See, carbon toxi, we had done the what was the toxi doing? Toxi was doing a relay. It was doing a relay through the chain, right? So in the normal toxi, what we saw, proton toxi, it was doing a relay through the proton chemical shifts. Now, but here you see, you do a carbon-carbon toxi, and carbon-carbon coupling is very large, about 35 to 40 hertz, the one bond coupling. Therefore, the mixing time required for the toxi is also not very large. So you can do it very effectively, do a toxi relay, and then of course you come back to the proton for detection. Okay, so you can do individual residue wise, you can do a carbon-carbon toxi relay. Okay, and that will allow you to identify the spin systems of the individual residues much better than what you would do from the proton-proton experiments. So this is an illustration here. A particular proton, there is a particular any residue, some residue which we don't know. There is the C delta chemical shift here, and on here you have the protons. Okay, so from the alpha proton, this is the alpha proton there. From the alpha proton, you go to the beta proton, to the beta the gamma, beta 2, delta 2, and delta 1. Okay. So this is at a particular, and the C alpha chemical shift is 52.6 ppm. Okay. And so therefore you have a complete relay of a particular residue. So on the basis of this relay, you can identify what that residue must be. Okay, because the chemical shifts are indicative of that one. Similarly, if you take this residue, you have the alpha to the gamma to the beta 1 and beta 1 is here close, beta 2, delta 1 and delta 2. These are typically indicative of methyl bearing residues because the methyl bearing residues appear in this area on the chemical shift. On the proton, chemical shift is quite upfield. So on the basis of that, you can identify those. See, all of these are delta here. So these are the methyl bearing residues. You also have deltas here. These also are methyl bearing residues. But the betas and the gammas, these are methylene protons. Therefore, this will allow you to walk through, identify the individual amino acids on the basis of the C alpha chemical shift region. This is the toxic spectrum of the C alpha. Okay. Now I talked about the, how many more are here?
How many slides?
3 more are there. Oh, 4 more are there. So we take a break?
I think
We take a break?
yes, yeah.
Uh-huh.
Short break, then
Yeah, short break and then we, yeah, take a break and then we'll come back. And after that, we'll Okay. So, no, previous one. Okay. So now we come to the next step of structure calculation. We talked about the structure calculation using NOEs, and we talked about the structure calculation using the chemical shifts. Now there is another important input which we also discussed earlier, which is called as the residual dipolar coupling. Residual dipolar couplings, these are obtained by aligning and putting the solution molecules in partially aligned media. And this residual dipolar coupling has the information about the orientation of the dipolar vector between the two things. Let us say two NH, and what is the orientation of this vector connecting the two things with respect to the magnetic field. So that is the angle theta, if you remember, that is partially average, and therefore, since it is partially average, it generates a reduced dipolar coupling. Now we introduce this reduced dipolar coupling as a parameter for orienting domains in the protein, which are sometimes not very well defined. So if you have a long range interaction, because the orientation gives you a long range interaction, a reduced dipolar coupling gives you a long range interaction, and that will allow you to refine the structure, especially when you have to orient two independent domains of the protein in a particular manner. Here is an illustration, for example. There is a long loop in between. There is a one domain here and another domain here, and the relative orientations of this is not very well defined. Similarly, in this case, the protein is one domain here, another domain here. Between these two, there is a long loop which is flexible, and one cannot define these with respect to each other properly. Same happens in many of these places. So here, this indicates, typically if you measure the RDCs, you measure the RDCs observed, and you plot them here with respect to what is your thing from the structure what we have initially. It is looking like that. So you have not optimized the structure with respect to that. But when you optimize the structure with respect to the observed RDCs, then you will see how the things start converging there. They all become calculated RDCs, and these RDCs are becoming the same. So this is the same thing here actually. It is at different levels. Different steps of the refinement. This is to begin with initial structure what we had, and now you refine it using the RDCs to a certain extent. How many RDCs you put, depending upon how many RDCs you have a certain set of agreement. Now you see, you put in all the RDCs, you come to this structure, this final structure, you see everything is perfectly fitting. The orientations of the two domains is perfectly satisfied. Therefore, RDCs reflect upon the orientations of the domains. If the orientation of the domains are good, then your calculated RDCs and the measured RDCs will be identical. So therefore, on one side you have the observed RDCs, other side you have the calculated RDCs, and when you have a larger number of RDCs, smaller number of RDCs here, certain sections of the things are used in this one. Here you're using all of them and everything is perfectly matching. Then you see the structure calculated, the orientations of the domains are perfectly defined. So this is the refinement process of the structure calculation, either you obtain from the NOEs or from the chemical shift information. Okay, now there is a third one, which is also we mentioned earlier, so-called the paramagnetic relaxation enhancement. This is again an input which gives you long range interactions. See, the NOEs go to 5 angstroms, but the PRE goes to 15 to 25 angstrom distance. So therefore, what you do here is you introduce a spin level, mostly hydroxide spin level. Typically, you attach it to a cysteine residue because the cysteine has an SH group. To add the SH group, you oxidize it and attach a spin level there. And spin level has a unpaired electron, and it has a large magnetic moment, and therefore it influences the protons at faraway places. Okay, therefore it influences the relaxation of the residues at faraway places. So they have a major effect on the NMR spectrum. So here are the 4 different situations there. The residues, here in a particular protein, you consider a D292C mutation. You introduce a cysteine at the particular residue, and at that point you attach a spin label, and that is here. This is here. This is the N terminal, C terminal, and this is a much larger protein. You can see here 292 residues. Normally in the NOE spectra alone, this is very difficult to obtain complete structural analysis. So you put a label at this point at 292 residue, and you are measuring the intensities of the peaks of the protein, certain section of the protein, in the presence of the label and in the absence of the label. Okay, and where this is, the ones which are coming under the influence of this label, those lines get broadened because it causes the relaxation enhancement. Relaxation enhancement meaning the R2s get enhanced, and therefore you have low intensities of the peaks. And that is why what is plotted here is the intensity ratio of the paramagnetic system and the diamagnetic system. The diamagnetic means when there is no spin level. Paramagnetic means when there is a spin level. You take the intensity ratios. The label is kept at one place, and you see these residues are getting perturbed. All these residues are so much intensities are reduced, other places it is not reduced. What does it mean? It means that these residues are somehow coming closer to the spin label. Wherever you put the spin label, it is coming closer. Now you put this label at different positions in the polypeptide chain, different portions of the spectrum are affected. If you put it here, this portion of the spectrum is affected. These residues are affected. If you put it at D56C here, at this position you get, then these residues are affected. If you put it here at 215, then these residues are affected. The red indicates that is where the effect is maximally seen. And if you put it at 201C, that is here, then these residues are affected. You see, so therefore you can see the combination of all of these will tell you which residues are coming close by. This also provides you a structural constraint. It provides you a structural input because you remember from the PRE you can calculate the distance. You can calculate the R, which is the inverse 6 power depending upon the PRE enhancement. So putting in these constraints, then you can actually refine the structure of the molecule further. So this is the use of PRE for structure analysis. PRE can also be used for identification of interactions. But with regard to the structure, this is the data which I got it from the PDV. How many NMR structures have been determined using of the different proteins? You see, in the initial years, 1989 to very, very few, hardly one per year, sometimes none. Okay, and this is where it was mostly NOE-based experiments. And as you came closer to the 1990s, then you started using heteronuclear experiments. When the heteronuclear experiments, the rapidity of structure determination increased, and then it increased quite rapidly. So now you see per year how many are getting determined. Quite substantial large number, and eventually it has now become this many. About 14,000 structures are in the PDV which have been determined. So this is the cumulative number. All this what I'm showing you is the cumulative number, how much are there as you have grown from here to here to here. So the number of structures has increased very rapidly, and this is because of the use of the heteronuclear experiments and multidimensional NMR experiments. Now this slide shows the PRE, use of PRE for studying interaction between proteins. Suppose I have, this is a particular protein, and this is the N15 labeled, okay? And you have this spin label is here, so it influences certain portion of the protein. Okay, this is typically used for studying Alzheimer's and all of those ones. Yeah, so this particular portion of the protein, some residues of these ones are affected. Here, for example, one consider the 36E. Now you make another one, you take a N14 labeled residue, not N15 labeled protein. You attach the label to the N14 labeled protein. This is the natural, it's not N15 enriched. In this case, you are attaching the MTSL, the spin label, to the N15 labeled protein. In this one, you are attaching it to the, not the labeled protein. Therefore, the signals what you will get will not be from here, you will get it from this protein, right? Because you are getting an HSQC spectrum. HSQC spectrum is proton to the nitrogen 15. You are getting signals of this one. And in this case also you are getting the signals from this one. So what is the difference here? Here, if the two proteins are interacting, so you are seeing which region of the protein is interacting. You have the label attached on a protein which is not N15 labeled. So therefore, this is the diamagnetic system. Okay, this is a diamagnetic system and this is a paramagnetic system. Because what you are measuring is the N15 is the spin label is attached on the N15 itself. Here the spin label is attached on the nitrogen 14. Therefore, the one what you are measuring is a diamagnetic system. Okay, so therefore you can see the intensities of the peaks if it is getting affected here at all. If it is getting affected, that means that okay, these two are interacting, and which residues are getting affected will be seen by these intensity ratios. So you plot this in normalized peak intensities here for 36E, for example. So the amino acid residue 36E, you put the label somewhere, and that is showing you a paramagnetic one is here, but the intensities have gone down, intense because of the paramagnetism. And the diamagnetic one is here. So this decrease in the intensity you can use for calculation of the interactions. And these are the spectra here. Okay, the spectra of this you can see how much is the change. Okay, so now you can see this, and this is the relaxation rates. The relaxation rate, this is telling you about the decay. So the decay therefore is with respect to the time, right? So this is therefore against the time. Peak intensities are decreasing with the time. So how it is T2 measurements, peak intensity, how it is changing. That will give you the relaxation rates. See, proton R2 relaxation rates E36 amide, which locates close to the interface of the two proteins. Solid lines represent fits to single exponential. You fit them to single exponential to calculate the R2 values. From the R2 values, you calculate the difference will give you the paramagnetic RE contribution. From that, you calculate the distance of this from the paramagnetic spin level, and that is how you calculate the interactions between the two, especially when you have this. So this is another protein there for interaction between two different proteins. Okay, so with that we conclude this section, and we have seen the various ways how one can calculate the structure of the folded proteins, and we calculate the interactions between folded proteins. Now we go over to more challenging systems. Fine. Yeah. Okay, so you go over to this new set of class of proteins which are called as intrinsically disordered proteins or intrinsically disordered spectral regions, IDPs or IDPRs. I mentioned this before when I talked about the general introduction to the protein world, that these proteins are becoming now important from the point of view of understanding biological functions. These are very flexible, no definite structure, may adopt different structures on binding to different partners. And binding is a very common thing in biology, right? So they will have to bind to different partners. And IDPRs mean there are certain sections of the proteins, part of the protein is folded, but some portions of the proteins are not folded. There's a loop areas, for example, and those are called as IDPRs. And this is a common feature of both of those. Now, such proteins cannot be studied by X-ray crystallography or by cryo-EM because they are flexible. Since they are flexible, you cannot obtain crystals of these proteins. And once you don't have the crystal, you cannot determine the structure. And many of these ones are small, or quite small for cryo-EM. Cryo-EM works very well when your protein system is very large. So let's say 500 amino acid residues, things like that, 600, 1000 amino acid residues. Then the cryo-EM works very well. But if there are smaller proteins like 150, 200 amino acid residues, which contains such disorder regions, flexible regions, then they are not suitable for cryo-EM. So in this situation, NMR becomes the only technique whereby you can obtain some information about the characteristics of the protein. Are there any challenges from the NMR point of view? We will see. So here is a comparison of a protein which is folded and unfolded. So ADPs belong here. Okay, and this is the folded protein. It's the same protein which is unfolded. Okay, and what is the consequence of that? You see the chemical shift dispersion which is present here from here to here is reduced to only this much. The amide proton chemical shift is completely eliminated because of the unfolded region. I showed you this in the very first slide also. Unfolded and folded proteins, the chemical shift dispersion in the amide protons gets lost, and that is reflected here. Okay, so therefore this becomes a challenge too for NMR. Fortunately, however, the N15 chemical shift, see N15 chemical shift from here to here, here to here, this is the same. This is not very different. Okay, the N15 chemical shift doesn't change much, and that is actually a positive point. One can make use of this N15 chemical shift to design experiments so that all these peaks can get distributed in different planes of the three-dimensional experiments. Then you can start analyzing those spectra. So you have to design pulse sequences for that, design experiments for that. Okay, that has been a big effort. And here I show you two pulse sequences which were developed in our laboratory. Several other people also made certain other efforts, but there are some distinct advantages in these ones. Therefore, I'm showing only this. We will not have much time to discuss any other ones. So here is an experimental, there are two experimental sequences which are listed here. These are called as HNN and HMCN. And in both cases, the schematic of the spectrum is like this. You have the amide proton on one axis, and on the other two axes of the three-dimensional spectrum, you have a nitrogen 15. Nitrogen 15 along both axes. Okay. However, the way the magnetization is transferred is different in the two cases. Okay. That gives you a different kind of information. But the N15 chemical shifts are present along both axes. Therefore, this is a big advantage because we have made use of the N15 chemical shift dispersion along two of the three axes. This dispersion is very small, we know. But therefore, to circumvent this problem only, we used N15 chemical shifts along two different axes. Okay, so here you notice this, we will not go to the details of the pulse sequence there, except I would like to notice some of these numbers here. These are variables called tau C, tau Cn, and because these will appear in the certain of the equations which are there. The magnetization transfer I will describe. The pulse sequences are designed according to the magnetization transfer pathways we want. Okay, and in those cases, in those ones, these parameters like tau C, tau Cn, and tau Cn appears in both cases. These appear in those equations there. So those ones are sufficient to understand the behaviors of the experiment. Okay, let's go and start looking at what is an HNN pulse sequence. The HNN pulse sequence, this is the polypeptide chain here, from the left hand to the right hand. This is the N chain, N terminal, and C terminal. The residue that indicated I minus 1, I and I plus 1. Now what you do here is the residue number I. From residue I, you transfer the magnetization to the nitrogen 15 here. From nitrogen 15, this transfer, and you label there T1. First you label T1. Then from the nitrogen 15, you transfer to the C alpha here as well as here, as in the case of HNCA. Now from this C alpha and the C alpha, you transfer to the next nitrogen. Additional step is involved here. This is the important part. You transfer to the next nitrogen and you label this here T2. You label this T2, okay, and then you go to the nitrogen 15 here. The amide proton here, this is T3. And from here, the same way here, from the C alpha, you go to this nitrogen. And from this, when you are transferring here, you also transfer back here to this nitrogen. Same here, from transfer back to this nitrogen. From this nitrogen, you go to the amide proton. Therefore, when you are labeling the nitrogen, you label this one in T2, this one in T2, and this one in T2. Okay, all the three nitrogens are labeled in the T2 period. In the T1 period, this nitrogen alone is labeled. So this nitrogen appears twice, once along T1 and once along T2. This appears only in T2, and amide protons, all these three appear in T3. What is the consequence of this? Let's see that here explicitly. Oops. The same thing I've explained to you in a little bit more clear manner. So you start from the amide proton, transfer to the nitrogen Ni, and you label the nitrogen as T1 there. From here you transfer to the C alpha and C alpha I minus 1, C alpha I and C alpha. This is the constant time period which is there, that will appear in the equations. C alpha I to C alpha I minus 1, and from C alpha I minus 1 you transfer to the nitrogen I plus 1, nitrogen I, self residue, and from C alpha I minus 1 you transfer to the I and also to the I minus 1 of nitrogen. And all these are now labeled in T2. These nitrogens are labeled in T2. Okay, and then from this nitrogen you transfer to the amide proton, which is labeled in T3. From here you go to the amide, from here you go to the amide. So what do you expect? How you should expect your spectrum? Oh, okay, this additional W2 F2 is there. That one should delete it. Anyway, that doesn't matter. So what do you expect here? So this is a particular value of nitrogen. Let's say amide proton nitrogen I, F1 is equal to Ni, because I'm looking at this only. This is T1 means F1 will be Ni. F1 will be Ni chemical shift. And F3 will be what? Amide proton. And F2 is also nitrogen 15. Therefore I'm looking at the F3, F2. If I look at F3, F2, I should see these three, right? This and this, this and this, and this and this. So these three peaks I am seeing here in this. Okay? HNI minus 1 to Ni minus 1, HNI to Ni, and HNI plus 1 to Ni plus 1. These are the correlations we will see, and that is appears in the HSQC spectrum like this. This is a typical HSQC spectrum, right? So three residues are sequentially connected residues are picked up in this one particular experiment at the particular value of Ni. Now supposing I write this, it's starting from HNI. What I do, I write it in starting from two other residues, HNI minus 1, and then of course HNI plus 1. Let's look at HNI minus 1. Instead of starting from HNI, I start from HNI minus 1, let's say. Then what do I go? I go to Ni minus 1, this label as T1. Then I go to C alpha of I, like here, C alpha of I minus 1 and C alpha of I. Okay, Ni minus 1 to C alpha of I minus 1, that is the same. This one is the same. And this one is this one here. C alpha, right. Okay, Ni minus 1 to C alpha of I minus 1 and C alpha of I, right. Did I make a mistake there? This is correct, no? Ni 1 and the same and 1 at the lower one. Yeah, same and 1. Ni minus 1, I took one higher here, is it? Ni plus 1, it should be the same, it should go in the same manner. HNI minus 1 to Ni minus 1. Let's see here. Let's say this is HNI minus 1 to Ni minus 1, Ni to C alpha. Okay, now there is a problem here. This is not right. See, Ni minus 1 to C alpha I minus 1, which is the same as this, and this should be 1 down. C alpha I minus 2, it should be 1 down. Then C alpha I minus 2, C alpha I to Ni minus 1, and then it goes C alpha I to Ni, C alpha I minus 2, this will be Ni minus 2. क्या हो गया? I should see. See, Ni minus 1 to C alpha I minus 1, and then goes to the other side, I minus 2. The C alpha I minus 2, I should go to Ni minus N, okay, Ni minus this, okay, this is good, that this is wrong here. This will change it, Ni minus 1. So, and then similarly from C alpha I minus 1, I go to, this goes to Ni. I started from then C alpha to Ni minus 1. C alpha I minus 1 to Ni, C alpha I minus 1 to Ni minus 1. Okay, this is correct. So this is fine. So C alpha I minus 2 to N I minus where did it go? Ni minus 1 to C alpha I, Ni minus 1 to C alpha I minus 1, and Ni minus 1 to the next one will be, which one is higher? Ni minus 1 to C alpha I minus 1, Ni to Ci, that is this, and this will be 1 down, to 1 down. So therefore this is I minus 1, this is I minus 2, this is correct. Okay, now C alpha I minus 1, where I will go? From C alpha I minus 1, I will go to Ni, this is right, and C alpha I minus 1, I go to Ni minus 1, 2, this is right. Now C alpha I minus 1, where do I go? C alpha I minus 2, so I should go 1 up and the same. So I minus 2 and I minus 1, this will be I minus 1. So this will appear, there I will get an Ni and an Ni minus 1. There will be 2 here, Ni minus 1 and Ni. Ni minus 1 will appear twice. And there is another. So this, there is a mistake there. Huh? So this will have to be corrected. Okay. So I minus 2 to Ni, it's exactly the same pattern one should go. So this, the one goes up and comes down. So C alpha I minus 2, it goes to Ni minus 2 and C, and then also to Ni minus 1. Same and 1 less. And C alpha I minus 1 goes to Ni minus 1 and Ni. So there will be 2 here. So therefore Ni will come there, and also there will be Ni minus 1 will appear twice. Okay, so similarly one can do that here as well. So if I start from Hni plus 1, I go to Ni plus 1. Ni plus 1 gives me C alpha I plus 1 and C alpha I. Ni plus 1 to C alpha I and C alpha I plus 1. This is correct. So same residue and 1 residue behind. And same residue, 1 residue behind. And this, from here I go to C alpha I plus 1 to Ni plus 2, which is correct. Then C alpha I plus 1 to Ni plus 1 its own. And here I will get C alpha I to Ni its own residue and own residue, and Ni, do I get Ni plus 1? C alpha I to Ni plus 1. Yeah, this is correct. So, yeah, this is correct. So, therefore, here you will see, this one you will write a little bit better. Huh? Hni minus 1 to Ni minus 1, and Ni minus 1, it goes to 1 higher, and Ni to C alpha I, Ni minus 1 to C alpha I minus 1, and then 1 lower, C alpha I minus 2. This is correct. Okay, from C alpha I minus 2, so from each one I got the same and 1 higher, the same and 1 higher. So therefore C alpha I minus 2, let me write here, Ni minus 2 and Ni 1 higher and 1 and the same. Okay, that is the same and 1 higher, that is Ni minus 1. So I get a Ni minus 1 there and another Ni. The Ni is coming from here, Ni minus 1 is coming from here. So then I will have here Hni minus 1 will appear twice. Okay. So from here, Ni minus 1, this Ni minus 2, this will give me HNI minus 2. And HNI minus 1. These three we will get. Okay. So therefore, when I see this at particular NH of I, how many times HNI will appear? HNI is appearing here. So therefore, it gives me Ni plus 1. HNI is appearing here. It gives me Ni minus 1. HNI is appearing here. It gives me Ni. Therefore, at a particular HNI, I see to the N15 of I minus 1 and I plus 1. So these three you will get in the same line. At the NH of I, you see to the I. Why I am getting this? Because NH of I see the how many times it is appearing? Hn of I, it is appearing to N because of the T1 here. This is the F1 and this is F3. So I should see what is present here. What I see here and what I see here. So HNI is giving me an Ni minus 1, HNI is giving me an Ni plus 1, and HNI is giving me a Ni. So these three are present in this vertical line. So therefore, from this F1, F3 plane, you see the sequentially neighboring N15 chemical shifts quite clearly. So this is an extremely useful thing for going through that. And schematically, once you write all of these things, this can be derived. Okay, is that clear here? Or should I write it explicitly? More clearly? Should I write it explicitly? This is clear? The way you go, how the transfer is happening? One residue higher, one residue up.
Clear.
Clear?
This is maximum.
Okay. So, and now after that you make a quantitative calculations of all of these steps. You calculate the intensities of the peaks on the basis of these various transfer processes. And these are complex equations here, and we need not run into that. These are dependent on various coupling constants, 1 bond, 2 bond, and the C alpha C beta. This is the crucial one. C alpha C beta coupling is a very crucial one. And in other words, because this determines the signs of the peaks. And you notice here in the previous slide that I have this negative red and black. The red and black actually, on the one hand, they are indicating the sequential peaks. On the other hand, they are also indicating the sign of the peaks. The self peak is positive and the two sequential peaks are negative. More for most residues, except in the case when there is a glycine. Why? That is because of this coupling constant there. The coupling constant C alpha C beta is not there in the case of glycine. Therefore, anything which is neighboring to the glycine will get affected by this. Whether it is on the I minus 1, especially on the I minus 1 side, either on the I side or the I minus 1 side gets affected. So this is, once again, I put the same thing in a schematic manner here. So this spectrum I already explained, this also I explained to you, and this is called as the triplet filter through the HSQC. This one, if you see, it is like a triplet filter through the HSQC. This is the F2, F3 plane, and this is the F1, F3 plane. F1, F3, amide proton is along the F3 axis, and the N15s are along the F1 and the F2 axis. The F2, F3 plane gives you a triplet filter through the HSQC, and F1, F3 gives you the sequential connections between the three neighboring residues, amide proton to the three nitrogens. Okay, the next experiment is the HMCN experiment. This also goes in the same manner. You start from the amide proton to the nitrogen. Now you don't transfer to the C alpha directly, but you transfer to the carbonyl. This is like HNCO-CA. Okay, transfer to the carbonyl. From carbonyl you go to C alpha, and when you transfer from C alpha, now you transfer to the nitrogen. You transfer to this nitrogen and this nitrogen. Okay, and then you from the nitrogen you transfer to the amide proton here and also here. So therefore you don't go to the I plus 1 residue here at all. You are between I and I minus 1. And this provides a directionality. HNN did not provide you directionality. It went in both directions. Here it provides you directionality because you are going through the carbonyl. Just as it did in the case of HNCO-CA. The moment you go through the carbonyl, it provides you directionality. That is why we say here H and C, the C is put in the center, in the bracket. So you are not frequency labeling the carbonyl. You are frequency labeling only the nitrogens, but you use the carbonyl through the pathway, and therefore you still require for this doubly labeled proteins, N15 and C13 labeled proteins, because you are using the C alphas and the carbonyls for the transfer process. Okay, and see, as you said, you're using the C alpha and CO, but they are not frequency labeled at all. So the transfer process here is indicated in this manner here: HNI to NI, NI to C alpha, C dash I minus 1, C dash I minus 1 to C alpha I minus 1. Now C alpha I minus 1, you go to two of these, NI minus 1 and NI T2. This is labeled here. This is the frequency labeled here. And then from these you go to the respective amide proton to measure it. In the same manner, you can also write, as I wrote in the case of starting from HNI minus 1 and HNI plus 1, you can write the same thing here as well. And when you do that, you will get this information here. This is similar in the case where there are 3 peaks. In this case, you are seeing 2 peaks, I and I minus 1. But you will see in the F1, F3 plane, you actually see correlation to the I and the I plus 1. F1, F3 plane, that is this plane here in the 3-dimensional spectrum. You take a cross section through this here, and you will see in the thing you see I to I plus 1 only in this one. Okay, and here you see I to I minus 1. Therefore, you are not lost any information. You have this full information present on this, but it provides you directionality. The advantage here is that you directly know which is your neighboring N15. Remember in the case of HMCO, which I explained to you, you have to scan through the N15 planes to find out where the C alpha is, and that is a pain in itself. And you don't know which nitrogen plane to go. But here you know each nitrogen plane is your neighboring nitrogen plane. Therefore, you can directly go there and you can search through the C alphas for that if required. Okay, now I mentioned to you that the glycines play a very important role. The glycines, because of the C alpha C beta coupling. C alpha C beta coupling is not present in the glycines. The consequence of that will be, depending upon where the glycine is, all these sequential peaks have different kinds of sign patterns. For instance, here, this is the HNN, this is the HNCN here. For instance, here, this is the typical, these are the F1, F3 planes, by the way. Okay, these are F1, F3 planes. F1, F3 planes, what do we have? At the central amino acid residue, we have from residue I to I minus 1 and I plus 1. I minus 1 and I plus 1, correct? So, whereas in the case of F1, in the HNCN, I and I plus 1. So, those are the kinds of patterns what we will have. Now you see the kind of sign patterns what you get in your spectra. Okay, so this is the normal one which I showed you earlier. But now suppose there is a glycine at the residue I, YZZ. This is a triplet YZZ. So Y is at I minus 1, G is at I, and Z is I plus 1. Y and Z are residue other than glycine and proline. Other than glycine and proline. Because the proline doesn't have the amide proton. In this case, you will see the glycine, when it is in the middle, it is always negative. And the squares are indicating the diagonal peaks from the self peak. So I consider here 5 residue G, G dash, Y, X, Z. G, G dash are both glycines. Okay. So the G is always negative when it is in the middle. Okay. Now, if I have a Y, Z, Z, you see this diagonal is negative and it's sequential to the Y. Y is on the I minus 1 side. That is negative. But the sequential to the one which is on the I plus 1 position, Z, that is positive. If I take this GXZ, now the G is present at I minus 1 position. X is this one here. That is the diagonal. This is self. But this sequential to G is positive. This is not negative. Unlike here, this is positive. And the next sequential is negative. So you have 2 positives and 1 negative. Here, 1 positive, 2 negatives. If I go here, G dash ZZ, the diagonal is negative, but the sequential to the next glycine, this is positive. Okay, and on the I minus 1 side, G is negative. On the I minus 1 side, the Z is positive, and this on the other side is positive. PXZ, P of course doesn't produce your peak, therefore you have only XZ. Similarly, PGZ, G is negative, sequential is positive. YXP, because the P doesn't produce a peak. So therefore, that is in the same manner you can calculate that. Now in this case, YXZ, XZ, these are only I and I plus 1. Okay. YGZ, you see both these are positive because only I and I plus 1 AC. GXZ, both these are negative. X and Z, both are negative. G dash ZZ, therefore you're looking at G and Z. This is negative, this is positive. PXZ, positive, negative. PZZ, both positive. GZ is both positive, no matter what is on the other side. Therefore, these distinct patterns will tell you if you are walking along the polypeptide chain, these provide very distinct checkpoints. These are called as checkpoints. So I illustrate this to you with a particular sequence here. PGXZB. This is the sequence, a 5 residue pentapeptide sequence, and you have a G and there is a proline in this point. So you start from the G. The G empty circle, empty box is the negative peak, and the filled one is a positive peak. Now G to X, this is the sequential is negative, X is positive. Now you directly go to this N15 plane. You don't have to search through it. You go directly to this N15 plane and you will find a sequential self peak is here and which has a sequential peak to this one there. And then, okay, this is the this provides you the amide proton chemical shift. Okay, this one is the F1 F3 plane. This is the F2 F3 plane. You are not gone to the next plane yet. So from here you identify from the F2 F3 plane the amide proton chemical shift of this particular residue X. Now we go to the nitrogen 15 of this. In that one you see a positive peak at X at the same chemical shift as that one. And then you see sequential to this G, that must be this, and you find a sequential to this Z. These are sign patterns are very distinctly maintained. From here, Z sequential peak, you find the amide proton chemical shift from the F2, F3 plane there. There you get this nitrogen amide proton chemical shift. Then you go to that plane. Then you will see the diagonal peak here and the two sequential peaks there, X and B. Then you go to the B, and this manner you can continue. So therefore, by making use of the F1, F3 and the F2, F3 planes, you can identify the amide proton chemical shifts and the nitrogen chemical shifts very distinctly. This is shown here explicitly, writing only the F1, F3 planes, the same sequence, only the F1, F3 plane is indicated, F2, F3 planes are assumed because we already determined what is the amide proton chemical shift, we have already seen how to get it. Therefore, you can simply draw planes in this manner, G to X, X, Z to B. Okay, so all these peaks are present at the right place. So this is a sequential walk through the HNN spectrum. This is an experimental spectrum. This is such a beautiful spectrum of FKBP, and you walk, and this is only the F1, F3 connections. You go through the F1, F3 connections here. Okay, start from here, R71, Q70. Because there is a G on the left at 69 position, the patterns are different. You see, this is Q70 self peak, Q70 to 69. So you go 70 to 69, that is here. We are coming from the other direction. This line is drawn in the other direction. So you can go also here, 70 to 69 like this, 69 to 68, 68 to 67, 67 to 66, 66 to 65, 65 to 64, 64 to 63, 63 to 62, 62 to 61. You can walk either way. Which direction you want to go, your sequential peak and self peak can attach. And the peak patterns one has to remember clearly. And that is wherever there is a glycine, it causes a change in the peak pattern. Okay. You can see that already here in this situation here, the G62 is 62 to 61. The negative, the direction is negative, that is self peak. '62 to 63. So this can be clearly seen. Notice here you have two positives there. You have two positives. This is what we saw in the case of where otherwise typically you have the self peak is negative and the two sequential peaks are self peak is positive, two sequential peaks are negative. As it is as in these cases. These are like that. Self peak is positive and the two sequential peaks are negative. So there is 61, both 62 and 60 are negative. And when you go to 62, the self peak is negative, and you have two negatives and one this 60. So you go to 63. The 63 is now sequential peak is positive. Okay, so you go in this one. So therefore, you remember this positive negative peak patterns, and that is these are the checkpoints. This when you are walking through the polypeptide chain, so you must hit the right peak pattern when you are hitting the particular sequence at the residue numbers whenever there are the G. Similarly, this is another example where you can walk through the HNCN pulse sequence. HNCN pulse is this chain which is going from 35 to 50 here, the long chain which is shown there. Notice clearly the two positive peaks here, and immediately the neighboring ones are two negative peaks. This was what we saw in the schematic which I drew there, right? When there is a G, then you will have G, the two peaks are positive, and then you have the next one, you will have two negative ones immediately following that. Okay, so there is no other glycine anywhere. So only one glycine is present here. So just around that you have this kind of a pattern. Otherwise you have one positive, one negative, positive, negative, positive, negative, positive, negative, so on and so forth. So you see you can easily connect. A red connects to the black. Okay, then vertically, horizontally you go, red connects to the black. Vertically down also you can, black connects to the red. So in this manner you can walk through the polypeptide chain. Okay, so now other modifications in the pulse sequences which are possible, and so far in the previous ones we said okay we can make use of the glycines, and these provide you the checkpoints. The checkpoints in the glycines are very useful to walk through. Now it is possible also to generate other checkpoints, and that is called as the tuning of the HNN experiment. You can make the alanines also behave like glycines. Okay, or you can make the serines and threonines can make them behave like glycines because they have very distinct C beta chemical shifts, and the C beta you can apply selective pulses to the C beta chemical shifts because these chemicals are very distinct, and that is done by manipulating this pulse here in the middle. So I think we will not describe that in any mathematics of that in detail, but this is possible to do it, and as a result what you get here. So this is the modification of where the alanine is made to behave like a glycine. The glycine will remain as a glycine, all right, but the alanine is also made to behave like a glycine, and that is typically what you see here. In this situation, you see this is G63, F64, F62, and these are the same sequence. These are maintained. These ones are maintained. In the HNNA experiment also, these are maintained. But where there is a glycine, where there is an alanine, here is an alanine. Okay, A11 is alanine. Now 11 you see in this HNN A11 is appearing negative. In the normal HNN, A11 appears positive. The two sequential peaks appear negative. 11, 12 and 10, these appear negative. 12 and 10 appear negative. A11 appears positive. But in HNN alanine becomes like a glycine. Therefore, this will appear negative. Glycine will appear negative. And the sequential to that appears positive. See, notice the contrast here. Sequential to that on the I plus one side appears positive, and on the I minus one side it appears still negative. Therefore, this is the change in the, therefore the alanine also becomes a checkpoint. So if you are doing this experiment, modified experiment HNNA, you will get this kind of a peak pattern, and that will allow you to additional checkpoints along the polypeptide chain. Okay, so these, and then similarly here A21, A21. In the HNN, it's not affected. The HNN, the A21 is like that, and the D20 and V22, they are two sequential or negative. But in HNNA, you see the A21 has become negative, the sequential to the next one has become positive, the previous one is negative there. So these sort of patterns you can generate by these pulse sequences. You can do the same thing for serines and threonines, and these are the patterns which are indicated. This is possible. This is all published in these places there. You can, I mean, we will describe everything in detail here, but these are the experiments which are extremely useful to generate checkpoints, and in a simple way you can obtain assignments of the individual residues in a very rapid manner. This is the pulse sequence for that. For the HNCN-ST, this was HNN-ST, HNN and ST modification. Here is the pulse sequence for the HNCN-ST. You can modify, get serine threonine checkpoints here as well. Okay. So finally, I think after this we will stop. So you can take projections of this HNCN sort of experiments. Okay. And there are various HNCN sort of experiments where they generate 2D projections. A particularly notable one is this one and this one. This is a sequential walk through that one. If you take the F2, F3 projection, F2, F3 plane, that means you take a projection down the F1 axis. F1 axis is nitrogen. If you take a projection down the F1 axis, then what you get? You get only peaks of the glycines and the next residues. G47, K48, G10, E11. These are HSQC spectra, remember that. In the HSQC spectrum, I get only these. If I take a projection, record the HNCN experiment and take projection down the F1 axis, I identify only the Gs and the residues next to them. G75, G76, G10, 11, R54 is additionally coming here. There is another one which is 53 that is not shown there. Okay. Now, if I take the projection down the F2 axis, then what I will get the F1, F3 plane. In the F1, F3 plane, I get all the HSQC peaks, black ones. All the black ones are the HSQC peaks. And I get additionally the sequential connections to those. Sequential connections to each one of those residues. And they will all become red. So therefore, I will have twice the number of peaks here, and in the same plane, I get the self peaks as well as the sequential peaks. Therefore, I can walk simply through this experiment, through this plane, and you can see that here, you can walk through this plane, make sequential connections, red to black to red, red to black, black, and now it is the G. Because there is a G, these two both are black. Okay, then therefore the next one immediately both are red, and therefore these are checkpoints. These checkpoints, this will allow you from here you go to again back to red, black and red, black and red. So you can make a sequential walk through this one 2D plane. And how long does it take to record such a spectrum? When I take projections mean, I actually don't have to increment that time variable. If I say F1 projection, which means I don't have to increment the T1 period, I just keep it fixed. So essentially I'm recording a 2D spectrum, which is equivalent to recording a projection, taking a projection down the F1 axis. In this case, I don't increment the F2, T2 period, therefore I get a 2D spectrum, which will be basically the F1, F3 projection of a 3D spectrum. Therefore, this can be recorded in half an hour. This experiment each of these can be recorded in half an hour. These 2D projections, now in this 2D projection, you are able to make a complete connection of the backbone. See from where to where you have come. It used to take from several months to years to obtain backbone assignments. Here you get it in one spectrum. In this projection, N15 and amide proton. Of course, after that you go to the other experiments to identify the other backbone protons. Okay, well, maybe I said I will stop after this.
Yeah, I think.
I'll stop. It's already quite heavy.
Yeah. That's what I wanted to say.
Yeah.
Heavy for them.
How many we did? Now how many?
Any doubt?
Any doubts are there?
Just cite any doubt? Supposedly, right, it's clear.
What I will do is next time I will correct that slide.
Replace that one.
I will replace that one. Replace that one.
Yeah. I think not many people are responding. Tapos responded, correct, clear. No? Very good. That is earlier one, first of. Okay, then you can,
Yeah.
can close.